Hello and welcome everyone to season 4 of Contact's Produce Safety webinar series. We've hit webinar 29. This series is part of a larger industry outreach and risk mitigation project called Contact, or scientific challenges and cost-effective management of risks associated with implementation of produce safety regulations. This project is supported by the Specialty Crops Research Initiative from the USDA National Institute of Food and Agriculture. Any opinions, findings, conclusions, or recommendations expressed in this presentation are those of the speakers and do not necessarily reflect the view of the U.S. Department of Agriculture. And we thank them for their funding. So, probably by now, you hopefully know who Contact is. But if not, for any of our new listeners, many universities and government institutions make up the Contact team. It's comprised of over 50 faculty, staff, students, and volunteers, not to mention the support of our Produce Advisory Committee. It takes a village. We're very appreciative of each individual's support and ongoing efforts to make Contact a grant champion by you all. Contact is now in its final year. It's hard to believe after about 5 years. So we are very grateful for your support, all of us. We wanted to bring your attention to a few resources, our Contact website. You can find a lot of the publications that will be discussed today, as well as some information on training programs and other grant findings. You can also visit our YouTube page. We house all of our video content on the YouTube page. It's got over 60 videos. If you want to be able to be notified when special things are dropped or like the recordings of these webinars are released, you can hit subscribe and follow along for notifications. And again, all really good content on there. We also have always done flyers, so you can see these are a list of some of the topics that we have covered. And again, they're all available free for you all to view at any time that you'd like. We also have our research summaries. These are located on our website. They're developed for each of the Contact research publications. They're freely available. We're also translating them into Spanish. They have a one-page PDF that links directly to the publication and provides a high-level overview of key results, as well as there's corresponding PowerPoint decks, very short, less than 10 slides, that also have slide notes that will allow you to be able to integrate some of the Contact findings into your outreach, education, and extension efforts or some of your presentations. And again, all freely available to use. And here is a list of some of those as well, but you can find those on the website. So that gets us to today's webinar. I want to remind everyone to submit your questions using the Q&A box. You can submit those at any time that you would like, and we will leave about the last 30 minutes to go through and ask those questions exactly how they show up, and we'll get into it. I'll start, but I'm joined also by Dr. Faith Kreitzer and Dr. Michelle Danilek, the University of Georgia and University of Florida. I'm sure they'll introduce themselves more. For those of you who don't know, Faith's been on the webinar series several times, and then Michelle is our fearless leader of Contact, and you've seen her as well. And then usually I do not actually do much talking beyond the Q&A, but this is a topic that I really enjoy, and so definitely wanted to jump into the hot seat. Dr. Laura Strong from Virginia Tech, and today I'll be setting the stage for post-harvest water practices, and more importantly, correcting some persistent dogma that we see in the field. So a quick shout out, of course, to a lot of the collaborators and support who make this work possible. One shout out to the Washington Coffee Group that recently hosted me during my travels for engaging in conversations related to this topic and being really open and honest, as well as our funding partners as well, and amazing industry. Again, this is always a team effort, conversation, and honestly, conflict and conversation push science and best practice forward. So why do we even care about this topic? Why is it top of mind? We've seen serious outbreaks tied to post-harvest water, everything from romaine and papaya to cantaloupe or mango. These cases, they weren't theoretical. These are real preventable illnesses tied to different parts of our water systems, including, you can see, from source water, recirculated batch, wash water, single pass, wash water, and even heating and cooling. So Definitely something that is an area, right, for amplification and spread if we're not careful. And so that's why, again, this webinar is definitely top of mind and the research is extremely pertinent. Post-harvest water is used everywhere, from dump tanks to flumes to spray tables, hydrocoolers. It removes dirt and debris. It's great for cooling, commodity movement. We use it also in our cleaning and sanitation activities and even hand washing. And each of these different kind of water systems that you can see on the screen come with their own inherent different risks and profiles. And this is really important that we can't really apply a one size fits all with rules. And it's really important that each operation look at how they're using water and go through this process. And hopefully, we'll give you a lot of tools and a lot of the current science to make decisions to best support your operation and the management of this water. But you can see we go from a low to a high, a lot of it based on the contact that it has with the produce in terms of the time, and some of it being recirculated or batch, which gets us into thinking about these systems in two broad types. There's really single pass and this recirculated or batch systems. And with a single pass system, this is going to be water that's used once and discarded, right? We're not using it over and over. So the focus is really on initial water quality. But with a batch or a recirculated system, you have to maintain the quality of that water over time. And that's where our sanitizers and things like change schedules are essential. You can see I've divided them up between small and large volumes of water, but certainly this will give you a little bit of a cheat sheet. To further that cheat sheet, looking at these two bigger systems, our single pass systems and our batch recirculated systems, again, we really want to focus in on the initial quality of water. That's always important no matter what we're doing and no matter what, if we're going with regulation or an audit. Then we really want to think about how we're using it for sanitation, how we're going to clean this type of equipment. What does it look like? For our recirculated and our batch, we're really going to focus in on maintaining the quality of the water. When we think about how to maintain the quality of that water, we're thinking of really two things that we have in our toolbox. When are we going to change the water? And our sanitizers. And for our single pass systems, you'll also see sanitizer use come up because of prevention of biofilms. So water just isn't a tool, right? But it can also be a vector for spreading potential microorganisms. The point here is that sanitizers prevent cross-contamination and are a control point in the system. But they're not miracle workers, so management of sanitizers and other relevant water parameters also matter, and you'll see that in the research and how it all ties together. So just as I think some of you have probably seen different animations talk about cross-contamination. So here we have some contamination, right? A contamination event. And we can't see this often, right? Sometimes you might be able to see visible contamination. That's pretty easy across the board, right? You're not going to harvest it. You're going to bypass that. It's where you can't really see that contamination, that where all this management on the harvest and the post-harvest side plays such an important role in terms of the spread. So we've got our tomatoes that we're harvesting and we're continuing to harvest, but we accidentally get some of that contamination. There are unforeseeable hazards that do happen that can occur. So what do we do, right? What are we trying to prevent here? We've got our dump or our recirculated system here. We're going to have our tomatoes go in. Maybe we're using this for movement, whatever. They're continuing to go through. Everything is fine, right? No cross-contamination. Our tomato that's contaminated goes through, and now we see that we have contamination in our wash water as well as on the tomato. And when we have other tomatoes go through, we're going to see that they now can pick up that contamination or it's cross-contaminated. And then everything else going through that water system can be cross-contaminated. And you can really start to see this spread of our contamination as well as potentially amplification if conditions are right. So let's look at what happens when you have a sanitizer. So we still have our tomatoes that we're harvesting. We've got that one bad actor that gets in, it's contaminated that we don't necessarily know. We have our system in place. We go ahead and run through the first couple. Everything's looking good. Have our next one go through. We have that contamination again. But with our sanitizer, you can see We get some reduction on the surface of our fruit, but can't say everything, right? It's not a kill step. And we also see, though, that we have completed inactivation in our water system. So that means that when our next tomatoes come through, they're not going to be contaminated. And really, another graphic, just here's the point. A system without sanitizer is risky, especially when pathogens are present. With a sanitizer and good practices, you reduce that cross-contamination risk. Hopefully that concept is clear, but this is one that, again, is just so vitally important to understand when it comes to management. So a reminder that washing produce, it's not a kill step, right? You can't wash off that contamination. You did see some reduction, and I think some of our speakers will talk about some log reductions, but we can't necessarily get that complete kill step. Our sanitizers maintain the quality of that water, but they're not going to necessarily fix that already contaminated product. So rework or rewashing, it won't change the outcome. We do have control points in our system, and we have food safety programs like our GAPs, our GMPs, and others to prevent spread and amplification. So when something does go wrong, the focus should be on reducing spread. That means biofilm control, facility hygiene, really breaking that chain of transmission. We don't want the contamination to be spread or amplified in the system. So sometimes the best option is not to wash at all. So if you're not going to be able to manage it or give the resources to it, we could do a dry pack, a field pack, or try to avoid submersion. The FSMA produce safety rule nor the GAP audits, they don't require that growers or packers wash produce. But if submersion produce is the only way, then sanitizers and water change schedules become non-negotiable in that management piece. There's a lot of different factors, but here's a non-exhaustive list of what you would think to manage or monitor. And you can customize this based on your operation. You don't have to track everything, but you do need to plan or make a plan that's tied to your specific systems and risk profiles for each water system. As I said, Faith and Michelle will discuss more of these in depth and what the research is saying. So I know, Faith, you'll hear a lot about chemical oxygen demand, and Michelle's going to talk some about contact times and sanitizers and whatnot, but we'll cover a wide variety of these. And again, all of them potentially could be relevant in your management strategy. So monitoring can be chemical, it can be physical, it can be observational. I think what matters the most here is that it's real time and it's accurate, or you have some sort of set frequency. Inaccurate measurements, especially sometimes with test strips that have become expired or been stored improperly, can lead to poor control and give you a false sense of security. Also, frequency is critical to establish how often you're going to monitor or verify these parameters, that we don't let our quality of our water drift in between checks. And again, some of the speakers will cover more on these topics. I want to spend a little bit of time before I turn it over to the research, just looking at sort of myth busting or demystifying maybe traditional norms that we see a lot. And I'll pick a few with temperature, turbidity, and ORP. But there are several references to outdated guidance and fact sheets that are still shaping industry practices, though I will say most industry wants to push back against some of these. And we need to push for updated science-based guidance and continued collaboration, again, to move beyond some of these outdated, maybe conventional norms that we used to see or that we still see. One of the first ones is around temperature or infiltration. And the idea here is that if hot produce is submerged or immersed in cold water, organisms on the surface or in the water can be drawn into the produce, and this is called infiltration. Doesn't happen the reverse way, but it happens when you have hot produce in cold water. And there's a lot of different guidance documents or fact sheets floating around. Myself, even, I used this one time in a document many years ago where we stated this 10 degrees rule. And I think it's important to know that really actually comes from a lot of plant pathogen research. It's not necessarily been established as the safe harbor on the foodborne pathogen side. And infiltration risks depend on so much more than just a straight 10 degrees, right, as a safe harbor. Instead, fruit damage, water depth, maturity, stem scars, time the produce spends in the water, all these things affect that risk of infiltration. And there's many practices that are used in industry to reduce this beyond just temperature control. You can avoid deep kinks, overloading, minimizing contact time, pre-cooling with alternative means, many other options here. I think it's that we need to reevaluate some of these standards in the context of our system-specific risks and be flexible and open to if someone's controlling infiltration without, say, 10 degrees Fahrenheit, what is it? And let's evaluate it based on the science. Another one is around turbidity and just concept of this 300 NTU that shows up a lot in guidance documents, or any really number, isn't a magic number. Turbidity can be used as a measurement of water quality. Yes, we learned that in the produce safety rule. And higher turbidity, the higher demand for sanitizer usually. But various food safety schemes can also require turbidity monitoring. And this may be an option, like I said, for understanding when to change your water, but it needs to be evaluated based on your system or the system in context. For example, in systems with robust sanitizer and pH control, there's data to support that turbidity often has little added value. So instead, we want to focus our resources and attentions on maintaining that sanitizer concentration, not necessarily the clarity of the water. I think it's really important to look at the individual systems and what you're doing. Also, just a shout out to some other work that's being funded by USDA. Dr. Claire Murphy has a project going on about in-field validation, packing house flume water, and looking at some of these parameters and how they play, and how do you set some of these base thresholds. Another one that I want to touch on is ORP, so oxidation reduction potential. Oxidizing can disrupt cell membranes and damage microorganisms. I think ORP is really great for automation, but it's important to remember that ORP doesn't necessarily reflect the concentration directly of your sanitizer. And you can see that on the right of the screen. We have this sort of stable ORP reading of about 800 millivolts, but we can see a pretty variable concentration of our sanitizer. And this is especially true as well in high organic loads. But again, it's going to be dependent on what you look at. So never want to say never. I think it's important to look at your data and look at your system. And also verify with direct measurements. I'd like to get away from just thinking, I have my ORP set, I'm good to go. I don't ever need to look at maybe checking that sanitizer concentration. In general, or in summary, there are many different types of post-harvest water systems that we use, each with different risks. You have to do the system-specific risk evaluation. Each system will have benefits and challenges. There's no one-size-fits-all. We need to evaluate each system with the following in mind, and this chart can help you start to get thinking and allow you to customize your water management plan, thinking through water source, commodity, sanitation, the water system, water treatment, workers, all these different things and other factors. We don't want to fall prey to dogma. We want to check that the science backs what we're doing. Just for an example, water hardness is under-recognized as an important parameter. It can also influence sanitizer demand in certain systems, but this is only going to be relevant based on water source and your system. So this lets you start to get thinking to customize, again, that water management plan based on some of the things like system type and audit needs. So with that, I will thank all of you for hanging with me. Hopefully this gave you a little bit of a crash course, and always happy to chat. And I'll tag in Michelle to get us started on looking at some of that spray bar data.
Perfect. Thanks, Laura. So I am going to pick up and touch on single pass water and talk through where the research is at on single pass water, different commodities, different systems, and where we're at. Wanted to start this with the discussion, building on what Laura said previously. Are we talking about adding sanitizer to water to prevent cross-contamination, which I think is what we always tell you in our trainings is the reason we're adding sanitizer to water, right? We want to prevent cross-contamination. We want to prevent in our single pass systems movement through the water to other produce, movement from the rollers onto other uncontaminated produce, or really prevent even movement from item to item within the produce. But we also get a log reduction. Some sort of a reduction happens when we look at the research and the data out there with these single pass systems. And there's this sort of interesting, not really a loophole, but an interesting sort of facet of post-harvest water when we talk about these single-pass systems that really allows us in some cases to look at this single-pass water and its ability to provide us with a log reduction. So I'm going to start there as we walk through this presentation. And so as we walk through this, as we start, within the produce safety rule itself, back in the version that was let out in 2018, when it was talking about mitigation measures, it included this idea that there's a way to use a mitigation measure to lower the amount of microorganisms on your produce to what might have been in the water originally. And it talked specifically in that rule about commercial washing. And that is what I've got in the photo in this picture down on the bottom left-hand part of the screen. In the rule we got last year, that implementation date was yesterday for the pre-harvest side. We talk about mitigation measures as well. Ooh, I moved forward and didn't mean to. We talk about mitigation measures as well, but in this case, in that 2024 rule, as a mitigation measure, it doesn't just talk about commercial washing. It actually talks about removal by other activities during or after harvest. And that's the photo down on the right where that language is in the rule. But what this really allows us to explore is the knowledge that we do get some sort of a log reduction during these commercial washing steps. And that is not coming from nowhere. That idea that there is some sort of a reduction during a commercial washing step actually has its origins back in the 2000 juice HACCP rule, so CFR 120. And within that rule, there's a very specific exemption for fresh citrus juice. It contains a bunch of nuances, but what it essentially is saying is that if you can achieve the 5 log reduction that's mandated by the rule in some sort of a cumulative way on the surface of that citrus fruit, then you don't have to pasteurize the juice afterwards. And so back in the late 1990s, Stephen Powell, who was working for the Florida Department of Citrus, actually published a bunch of work about the different types of log reductions you could get on citrus as it runs through different packing house processes that are common in citrus here in Florida. And really what he was able to find, and I've listed the 3 studies that really point this out, is that standard washing and rinsing with potable water, and that's in a published study in 1998, or a combination of that washing with some sort of a waxing can get you a log reduction. Now, neither of those things gets you to the 5 log reduction, but there are other things you can do to the surface of those oranges or while you're actually juicing the orange to get to that 5 log reduction. But this really is still in use today within the fresh orange juice industry as validations on how to achieve this 5 log reduction on the surface of a fruit before you juice it. And again, these papers, these references are still in use today. Now, back several years ago now, about 20 years after Steve did this work, the practices have changed in the industry, right? We all know that from the mid-90s to where we are today, in our fruit washing is very different in terms of different chemicals, different waxes. And so myself and my colleague Mark Reitenauer here at the University of Florida, went ahead and looked at some of those new practices to reduce microorganisms on the surface of fresh fruits and vegetables. Now, as we start this, the first thing we wanted to do was make sure that the E. coli Steve did all his work with back in the mid-90s actually worked the same as Salmonella did. And we did that in a laboratory setting. And I want to introduce you on this slide to this photo here on the right. This is our pilot scale brush washer system that actually fits inside a biosafety cabinet in our laboratory. It allows us to actually look at log reductions or cross-contamination on the surface of fruits or vegetables with using the actual pathogen and not being a surrogate. Steve didn't have this back in the 90s, but comparing E. coli, the E. coli that he used, the E. coli we would subsequently use when we went into the commercial scale or pilot scale production was the same as Salmonella. And we were able to do that. And in this case, the orange bar is the log reduction we saw with E. coli. The blue bar is a log reduction we saw with salmonella. And what you can see is that for a number of different sanitizers, detergents, even water, our log reductions of E. coli and salmonella were essentially the same. They were not statistically different. So we felt confident in the work that Steve did previously, and we felt confident going forward in our work that using this E. coli was a suitable surrogate for salmonella. Now, we wanted to then take the surrogate and look at what we would see with a bunch of different washing at a larger scale. So we went into 2 pilot citrus packing houses that we have in Florida. The one on the left is the one at the Citrus Research Center where I'm located. And the one on the right is at the Indian River Research Center where Mark right now, my colleague, is located. And we ran on those 2 lines 13 different types of treatments, and they're listed there, looking at how much log reduction we can get using these current chemicals on these different lines. And in this case, we did decide to use grapefruit, whereas Stephen had done all the work on oranges in the past. And if you go into this paper, we did pull all the data together from the 2 different centers, and we looked at not just the average or the median reduction, but we also wanted to look at a maximum and a minimum reduction across all the different trials. So in this table, what I've got highlighted by the orange boxes and what are bolded in the paper in the table are the minimum log reductions we saw across all the trials and all the lines. And so we ran 5 trials in each packing house. We ran 30 replicates in each trial, and then we ran 10 fruit in each replicate. So this is the average of 100, or this is the lowest log reduction across 150 different pieces of fruit in each different example. And so what you can see there is a log reduction, the minimum log reduction highlighted in those boxes. And so when we talk to people in Florida who want to make fresh orange juice and do a surface pasteurization of the oranges, it's those minimum log reductions we saw across all those trials that we recommend for them to use as their log reduction within their HACCP plan to get that 5 log cumulative reduction that they need. Now, interestingly, what I want to point out to you is those minimum log reductions, if we had the presence of any detergent or sanitizer in the water, gave us, in all cases, more than a 3 log reduction. And we feel really comfortable in recommending these to the Florida citrus industry for their use. And again, see it commonly used in fresh citrus juice, but also if we had any packing houses that wanted to use it as a mitigation measure, we'd feel confident in recommending it to them as well to document the efficacy of their commercial washes. So that's some of the data that exists out there in the citrus world. But data doesn't only exist in citrus. In fact, it also exists in tomatoes. And for this, we're going to start to tell this story to the Tomato Good Agricultural Practices, or TGAPS program that exists for round tomatoes grown here in the state of Florida. So within this regulation, which was supported and developed with the tomato industry and has been enforced by FDACS, the Florida Department of Ag and Consumer Services, since 2008, it actually includes language within this program about a 3 log reduction of salmonella on the surface of tomatoes. And so you can see here that it essentially says you can't peel pack a tomato unless you can achieve some sort of a 3 log reduction on the microorganism. As the document continues, it goes on, and it says, this is the way we want you to sanitize a tomato. And in this case, it specifically calls out sanitation of dump tank and flume tank water in these packing houses. And I wanted to pull this up as I gave this an example because this is really the first time we see that 10 degrees difference between pulp temp and fruit temp show up. And it's based on, like Laura mentioned, some plant pathogen data in tomatoes, but it's written into our TGAPS legislations. It's also where we see that pH being mentioned when we talk about free chlorine levels. And it's also where we see this maximum time for something allowed in a flume tank. So that's all held within this rule. But what this rule also says is if you don't want to put your tomatoes through a dump tank, and meet these different requirements. You can actually also use some other chemical as long as you can show through a reproducible scientific study that it can achieve a minimum 3 log reduction of salmonella or some other organisms. So again, this rule came into place in 2008. Enter Keith Schneider, my colleague at the University of Florida, who not only made my Gator background slide or background for me to use today based on our national championship win after night, but also was really active with the Florida Tomato Committee. And so starting in the early 2010s, Keith did a bunch of work looking at different ways to get 3 log reductions on the surface of tomatoes. And this is where that pilot brush system that fits inside our biosafety cabinet really started. It's a work he did with Alex, a master's student in his lab, looked at the log reduction you could get on the surface of tomatoes with different types of sanitizer. And so if we look at data coming out of this study, and we look specifically at this bottom graph here, this talks about the log reduction of salmonella on the surface of tomatoes at various times with 100 part per million free chlorine, 5 part per million chlorine dioxide, 80 part per million PAA, or just a water control. All right. And this, you can see that by 15 seconds of washing on the brush bed, they achieved an average 3 log reduction, any one of those sanitizers, and in fact, even the water. PAA did a little bit better than chlorine dioxide or free chlorine at 15 seconds, but that sort of at 60 seconds of washing, they all increased to the same spot. And so work Keith did and was published back in 2012. This was not the end of Keith's work on different overhead sanitizing brush roller systems. He then had a future student named Elena who did work looking at both spoilage organisms on salmonella using that brush bed overhead system and also the potential for it to damage fruit. And then he also did subsequent studies funded by a different SCRI that we had led by Bob Buchanan, looking at reductions in background populations on tomatoes in commercial facilities over multiple years in Florida. And then in between different systems, including flumes, overhead sprays, dry packing and field packing in packing houses in Florida, New Jersey, and in Maryland. And all showed very interesting results again in support of the fact that you could get a log reduction using an overhead single pass spray section. So within the Contact project, this work is ongoing. I have a student right now named Ashton who is repeating a lot of that early work that Alex did, looking at MS2 as a surrogate for norovirus on tomatoes. And the trends we're seeing on tomatoes are with MS2 are very different than what we're seeing within the bacterial pathogens. She's wrapping up this work now, plans to graduate this summer, and will be presenting it at IAFP. But essentially, if you look on these graphs here, and these are log reductions in the plaque-forming units of the bacteriophage, essentially at any different concentration of chlorine or PAA, PAA being in the blue and chlorine in the bottom, and this is washing for 30 seconds. Increasing sanitizer level did not get us increased log reductions of the different bacteriophage on the surface. So I've talked through tomatoes and I've talked through citrus. What work exists out in other commodities? There is work published out there on bell peppers. This is looking at a commercial packing house in Spain that's using PAA as their sanitizer, and this is out of Ana Allende's lab group. And what they found is that the pre-wash is a critical point affecting the microbiological quality of those bell peppers. The use of non-disinfected water, so if you've got water that doesn't have sanitizer in it, in that pre-wash step really increased food safety risks, but adding PAA into the process reduced the microbial load both in the wash water and on the bell peppers themselves. So we're again looking at increased importance of adding a sanitizer to that water. There's also work out of Jinru Chen's lab at UGA that was published back in 2021, and this is looking at various different packing houses, so at a commercial level, exploring a variety of different systems and sanitizers on peach quality. And in this case, they actually found in the majority of the cases that the fruit exiting the washer had a higher load of microorganisms than those entering the washer, and that they had a very high load of microorganisms in the spent peach wash water. And their conclusion was that they really needed to increase that concentration of the sanitizer within those peach washing systems. We've also seen work done on a smaller scale on blueberries. This is work out of Vivian Wu's lab at USDA ARS in Albany, California. And she was looking at overhead washing blueberries, the wild blueberries out of Maine. Again, in this case, specifically before freezing. They looked at a bunch of different combinations of free chlorine and chlorine dioxide, and they were able to use those two to reduce pathogens when compared to the control. However, higher concentrations of chlorine did not immediately lead to higher log reduction, so there was some nuance there. In this case, Vivian had set up a small system where they were overhead spraying into this tray of shaking blueberries in a biosafety cabinet in their lab. And if you check out the website associated with this paper published in 2023, they actually have a video that's pretty cool of what that system looked like. So other commodities that have been looked at include mangoes, and this was done out of my lab, and it's work funded by the National Mango Board in addition to the Center for Produce Safety, and completed by my student who finished with her MS degree several years ago, Zinue Wong. Here, we wanted to not only explore if sanitizers and washing times, but we also wanted to see if there was a difference between a brush roller and those PVC rollers we commonly see in these wash systems. And so you can look to see that we retrofitted our favorite brush washer that fits in our biosafety cabinet with PVC rollers. And in this case, we washed our fruit for different amounts of time, 5, 15, 30, and 60 seconds, with 100 ppm free chlorine, 80 part per million PAA, and 5 part per million chlorine dioxide. And we ran these all 5 times, each with 3 mango replicates per trial. Look quickly at the results here, and I'll point out really quickly, in this case, population. So these are populations, and the lines are decreasing. Up the x-axis and the y-axis is the time of washing. Blue lines are water, orange lines are chlorine, green lines are PAA, and purple lines are chlorine dioxide. And what we saw is that PAA worked very effectively on those brush rollers, almost to the limit of detection within 5 seconds. And when we look on our PVC rollers, we were able to see very similar results at 5 seconds for chlorine and for brush rollers, but by seconds of washing, significant reductions essentially across the board. We'll point out that our background population reductions were minimal in that study. So here we've got some more experimental data, and this is identical experimental design, except with the West Indian green skin avocados. And this was done by my former master's student, Lydia Valdez, who I saw on this call. And as of today, this work is still unpublished, but I really do hope to get it out in 2025. Again, looking at the exact same thing here. In this case, water is a dark blue line, chlorine is a purple line. PAA is the red line, and chlorine dioxide is a yellow line. And again, brush and PVC rollers. And what we can see is that after 60 seconds of washing, essentially we had good washing with everything on the brush except for the water, and with everything but the chlorine dioxide and the water on the PVC. But in general, the only place we saw really difference between the PVC and the brushes was with that chlorine dioxide level. And then at 5 seconds, wash with that chlorine dioxide, not quite as efficient as the chlorine or the chlorine dioxide. But in this case, on those brushes, any sanitizer was better than water. And on the PVC, any sanitizer other than chlorine dioxide did quite a bit better than the water. The other commodity where there's been a fair amount of work, all the way from surrogate validation, work on a pilot scale, and then through commercial washing, was out of Mei-Jun Zhu's lab at Washington State University, again supported by Produce Safety and the different associations up in the Pacific Northwest. And again, this work is complementary to the other studies that I've shared with you so far, again, speaking to the consistent reductions in the presence of certain levels of sanitizers. I also want to point out that with the exception of that early citrus and tomato work, all of this data has been published in the last year. So this is a very active field where a lot more work could be done. All right, so I spent all that time talking about log reduction, and now I'm going to talk about cross-contamination. And the reality is there is very little out there in the peer-reviewed literature about cross-contamination happening with this single-pass water. And so that's where contact comes in. So this is a study done by Dr. Mari Schroeder, who's a recent graduate with her PhD out of my lab. And one of her objectives was to look at cross-contamination again on our favorite pilot brush line and looking to see how things got spread in the presence of PAA and in free chlorine. So if we just set that system up here for you, we've got water coming in, going out of our spray bars. We used 3 tomatoes. We inoculated the tomato we put in the center. We didn't inoculate the tomato going on either side. And then at the end of our 30-second wash, we took both a sponge swab of those brush rollers, and we also enumerated salmonella in that wastewater itself. We inoculated at 10 to the 6 or 1 million salmonella cells per tomato. So no, that's a lot of salmonella there. And we washed always for 30 seconds in these experiments. So when there was no sanitizer present in that single pass water, we were always able to find quantifiable numbers of salmonella on all the different surfaces we looked at. So started with just under 1 million salmonella on those tomatoes. After we washed those tomatoes, there was 10 to the 3. So a couple log reduction we saw with that washing. But there was about statistically the same amount of salmonella, not only on that first tomato we washed, but also on those uninoculated tomatoes on either side of it. Same amount of salmonella we could pull off the brushes, and then just a little bit less salmonella we pulled out of the wastewater. So very similar salmonella on all of those different items. But when we added sanitizer, things started to look a little bit different. All right, so on this graph, on the previous graph, we actually had numbers going up the side. On this graph, what we've got is a percent number of positive surfaces or tomatoes going up off the side, because we could no longer count the number of cells that were present on those surfaces or tomatoes. So any sanitizer level there at all helped us not be able to count salmonella. You can see we increased free chlorine concentration, and when we hit 70 part per million free chlorine in that overhead wash, we could no longer find salmonella on the inoculated tomato, which was the red bar. The uninoculated tomatoes, which were the yellow and the brown bars, on those brush beds themselves with our sponge swabs or in our wastewater. So remember, we put 10 to the 6 salmonella on. That's 1 million salmonella. But once we hit 70 part per million free chlorine, we were no longer able to see it over there. We also have that for PAA. And in this case, looking at the same thing, percent positives, you can see higher percent positives at the 20 and 30 ppm PAA, but then pretty rapidly decreasing. And once we hit that 80 ppm PAA, we didn't see any cross-contamination happening. So as I wrap this up, 70 ppm free chlorine at 1 million salmonella on that one tomato, didn't see any cross-contamination. And once we hit 80 on PAA, again, we didn't see any cross-contamination there. So my takeaways for you is that, right, single pass water does not require the addition of sanitizers in our different regulations or most of our audit schemes. But in the absence of the sanitizer, we did see cross-contamination occurring 100% of the time to other tomatoes and to food contact surfaces in the study we did. For bacterial pathogens, we do also get a log reduction with those sanitizers, variable depending on the system we're looking at. And then the work with MS2 as a surrogate for norovirus is ongoing, and we hope to share that in the future. And I'll just quickly acknowledge the different funding sources for some of the data I showed, and then turn it over to Faith.
Great. Thanks, Michelle. Really appreciate it. I'm going to talk to you all today about post-harvest washing with these recirculated systems, where we definitely want to focus on some attributes. And COD is chemical oxygen demand. And we'll talk a little bit about why that is so important. So where are we going and why within this post-harvest recirculating world? I would say that all post-harvest systems are getting more and more scrutiny, and this is coming from audits, people evaluating your food safety plans or your HACCP plans. and really having a lot more questions raised than they have been historically in the past. They're really coming in and scrutinizing your operating parameters. And I would say, especially with post-harvest systems that are recirculated, this is critical in prevention of cross-contamination in these recycled systems. But as Michelle just showed, even in single-pass systems, there's good evidence where prudent use of these chemistries and these sanitizers really can help build up and eliminate cross-contamination risk. So, in thinking about this within the context, right, from you all that are part of the industry and looking at trying to have conversations with people around how you're managing your post-harvest water, these are the three things that I would split things into. The first is the question that you're trying to answer first and foremost. Unless you're really trying to go at it from the angle that Michelle just spoke about and look at log reductions on most commodities, and I would say that would best be done with the spray bar system where you have some abrasion also. Ultimately, the question that I'm asking usually if I'm thinking about an immersive type recycling system is how do you know the operating parameters are preventing cross contamination? That's going to be key and foremost because that's showing that you are preventing cross contamination, therefore preventing that risk factor. The first bucket comes into supporting documentation. So really look at information from peer-reviewed research, which will cover a couple of studies today to help justify your decisions. Sometimes you'll even find things from privately funded research, from commodity groups, or specific to your operation if you're doing things that are a little bit more unusual than what is typical standard for that industry. As Michelle just spoke about, this is a very active area of research. So I would say sometimes you're needing the answers before those answers are available to you. You also need to understand how your system operates. So what are your operational parameters? Think through what's critical in the description of your system because everybody's system differs significantly. So some things that we commonly think about here are sanitizer concentration, contact time with the product, range as well as the average, water temperature, range as well as the average, the chemical oxygen demand. For those of you who are like, this chemical oxygen demand, I've not really heard of this in the past. We think about it a lot with sanitizers, especially because they are oxidizers. And what a chemical oxygen demand test does is it will give you a number, a numerical value that tells you how much of that material is in that water in this instance that could be oxidized. This is never going to be a real-time measurement. We've tried to get at ways of estimating it better. But ultimately, this will take you 4 hours to get at this number. So it's not like it's something that you're going to use for routine monitoring, but it does help us describe these systems in a lot more depth than we would previously. Turbidity is another thing that we've looked at historically because it is more easily measured to look at that organic loading. Conductivity, pH, water hardness are all different attributes you could think through. And then the last bucket's really around any data around microbiological efficacy of the system that actually under your control and the way that you operate it. Just very briefly, the thought is here is that produce that leaves this wash system should not have greater microbial load than when it entered. And commonly, we see this conducted in plant with aerobic plate counts because that's where we have the highest microbial counts that we can achieve. Sometimes we can move to coliforms. Typically, and I would say this is a very good thing for the produce industry, we see E. coli counts that are so low to begin with, you're not really able to measure anything very readily. It's like you're too low of a population to really get good counts there. So what we're going to talk about in a little bit more detail today are these first two boxes. And know that there is some room to talk about through questions, that last one, if y'all want to ask some questions there. The first one I'm going to get into is the precursor to some of this work we did in these post-harvest systems that started when I was at Washington State. And this was funded through the Washington Tree Fruit Research Commission, and Corey Anderson was the student who conducted this research. Really, what we were trying to do here was to look at the water specifically. And our specific goal was really to understand the range and variability of different water quality parameters over time, and then to take that information and replicate it in the lab, actually working with foodborne pathogens in those water systems. So we recruited 3 different commercial packing houses that all manage their flume waters in different ways. There was one packing house that actually had a split flume. The thought process is there is that you dump the first flume more frequently, and the second flume, the organic matter doesn't build up to the same extent, so you can keep that water longer. We sampled every 4 hours up to 24 hours, and then we sampled every 12 hours until that water was changed. And we had people that went up to 5 days in that time period. And then we collected from these packing houses 3 times throughout the year. What we monitored were all those attributes I just listed previously, in addition to oxidation reduction potential. And ultimately here, we were just trying to get a pretty good data set that we could work from to look for both correlations on the hopeful side, and then if nothing else, to get some averages and ranges to describe these different water systems in this specific commodity. So amongst the 104 observations we had, we did not find any significant correlations between anything, right? That was really helpful in helping explain things. A few things that we did call out that I will talk about here is that ORP doesn't really make a whole lot of sense because we've averaged it in with the PAA and chlorine data. We did see much higher ORP values, of course, where they're using chlorine only. For pH, again, they're averaged in together here, but we did see evidence from the pH evaluation of the chlorine-only samples. The industry was doing a really good job of maintaining their pH in that sweet spot, about 6.5 to 7.5, so we get hypochlorous acid being produced. We also saw a pretty wide range of temperatures throughout this, with an average of about 70. Turbidity, notice that turbidity, we were never above 300 FAU, which is equivalent to NTU, and our average was 72. So we were getting into some turbid water at the end of this, but nothing above what's commonly referred to by others as that 300 cutoff. I will say I'm not a big lover of turbidity because it really doesn't help explain a lot of these compounds, but if it's a measurement that you've built your system around, just know that there may be a little bit of vulnerability with setting that as your standard. For COD, we did see quite a range here with an average of 592. And on the high side, we saw it up to 2510. For our actual compounds that they were being used, and I did split these out. So with PAA, we had an average of 62 parts per million. On the high side, 150, and on the lowest we saw was 2. For free chlorine, we saw far lower concentrations within these flumes, which I found interesting considering the cost. Overall, we had an 11.46 parts per million average, a high of 65, and a low of just recordable at 0.5. That was the lowest we could go in our titrations. When we look at the COD level, we know that this may be an attribute that not many people know for their own systems. So I wanted to give you a little bit of a feel for what did this data set look like. I've separated out by color each of these 4 different flumes. Remember, we did have one that was a split, like 2 discrete flumes within one packing house. Note that top bar up here so that we have our green line right here where we have an R square 0.46, not too bad R square. The reason that we see such a climb in turbidity here is that, not turbidity, but COD, was that we were seeing really the least amount of filtration within this system. All these other systems that you see don't get quite as high COD reached or range in COD is really given from the fact that they had other filtration systems in place. Some of them were using flocculants, others were using different size mesh filters that then went into more discrete filters. And even though they were refreshing that water at very low rates and replenishing it, it was still able to keep that COD level down fairly well compared to this system right here. Know that we also see that where we do sometimes have really good correlations, 0.46, 0.52, sometimes we're a little bit poorer, 0.15 and 0.2. So no, that means that basically even within 24 hours, if you're like, I would never hold my water past 24 hours, know that we can still see these COD values climbing quite significantly there. So if we went and looked at the data all together, we needed to get some basics around how are we going to set up our experimentation. So we went in and we said, okay, we're going to create a low COD value that's around 30. We're going to create a mid that's at 500, and then we're going to come in here and look at the uppermost limit and set that at 2,500. So we took these values into the lab and we tested them against 3 organisms, Salmonella, Shiga toxigenic E. coli, and Listeria, with those 3 COD targets. We looked at chlorine and peroxyacetic acid that I'm going to share with you all today. And then we looked at them over the course of 0, 15, 30, and 60 seconds. Just a few things to get us oriented with these graphs. Know that we have our time down here along our x-axis, our bacterial population, so that's population, so we want to see that going down. Within this waters right here is log CFU per milliliter, Know that something that's log 6 means that there's a million cells there. So it's the 1 and then how many number zeros come after it. Know that we also have Shiga toxigenic E. coli, Salmonella, and Listeria represented in white, yellow, and red bars here. I will separate those out as we move forward, but I thought for these purposes it was nice in seeing them together. If we talk about log reduction, a lot of times all we're doing is going in and taking the difference of where we started versus where we ended. And in this case, we started at 7.5 and we get down just by eyeballing it. You can tell you're just below 4. So right here, we have a 3.5 log reduction. And for a 3 log reduction, that's equivalent to a 99.9% inactivation. If it was a 2 log reduction, it would be a 99% inactivation. So if we actually look at the shapes of these curves, we can tell that free chlorine it reacts far more quickly than PAA does. Note that these are not a course of minutes. These are a matter of seconds, right? And 15 seconds was the fastest we could move and grab this sample and actually have it neutralized. So know that it could have been faster even. That's just the speed at which we could work. And when we go into other literature, we have seen evidence of this in the past, where free chlorine that's introduced to cells causes surface damage and cell lysis more rapidly. And that's why you see such a quick effect. And if it's going to work, it works very rapidly, and then it levels out and maintains similar inactivation levels. And so an increase in time doesn't really help you much there. Whereas with PAA, it really needed to actually migrate more into the cell, and it causes more intracellular oxidative stress by penetrating into those intact cells with longer reaction time compared to free chlorine. Know that we stopped at 60 seconds for a lot of your PAA products. They go for a longer contact time, 75 seconds upwards. And so know that time does matter more, especially with evidence like this. So I'm also going to walk through and show how free chlorine is really impacted by COD levels. And these are still our 3 organisms along different colors here. And what I want you to look at is this is our low COD concentration at 30 parts per million free chlorine. And now I'm going to overlay on top of it where on average we're getting somewhere between a 6 log to at least a 3 log inactivation with these organisms. When I increase the COD, so I'm going from a 30 milligrams per liter COD to 500 with our solid lines, you can tell that in every instance, I have reduced the amount of efficacy that I see within this water system and free chlorine. And I'm going to step it up once more to our high. And once more again, you see you have even less efficacy here. So many times we're taking, as we shift the COD higher and higher, you're doing a few things. From the bottom line standpoint, you're definitely impacting your economics because you're having to inject more of the compound to achieve more, whatever your desired set point is. And that usually helps people explain it enough. So it's all looking at water as an economy and how long do you have to hold that water versus how long can you refresh it and replenish it and recirculate that, because it definitely is driving efficacy as far as inactivating organisms here with chlorine. So with chlorine, this isn't too surprising, and hopefully this is not coming in new to anybody, but we have this chlorine demand that's driven by organic loading. We have combined chlorine, which many times doesn't change too much. That's the inorganic compounds that you're interacting with. And you have free chlorine, which is really our antimicrobial species that we're targeting. Everything you add is going to be your chlorine dosage. We know that when we increase that organic matter, and so if we're in the 2,500, let's say, range, we're going to see big increases in organic chlorine demand there. Ultimately, we're also going to see increases in combined chlorine and very little free chlorine remaining. And so we have to keep on adding more and more to overcome that. So that's our struggle with chlorine, and that's a well-known struggle that we all accept and interact with. When we look at PA, and this is a higher concentration of PA, and walking through similar things, first off, you'll see that the organisms, Listeria in our data set was far more resistant to inactivation by PAA than our gram-negative organisms of Salmonella and Shiga toxigenic E. coli. Know that as we step this up and look at, generally speaking, the exception of that Listeria data set, which is very reproducible, everything does step up in a similar stepwise fashion as we increase that COD level. And then if we increase to the highest, again, we're seeing a lot of times they would be very similar concentration or efficacy that we saw for our mid-range COD. So that's where when people are referring to not as great of an impact of COD, this is many times what they're picking up on and as far as the efficacy goes across the board. I'm going to break this down really briefly for you by organism. So I am not telling you all that a 99% inactivation or 2-log inactivation is what is appropriate. I just wanted to set a line in the sand so that you can easily compare these graphs. And if you go back and look to them on the webinar, you can look at these graphs. But in general, know that the results were very repetitive for what we saw across the board. Always just to anchor you, I'm going to have free chlorine right here in the blue box as we move through these. And we're going to have PAA in the green box. The PAA concentrations we held constant at each COD level, which are there on the side for you. But know that with chlorine, we had to keep on marching it up as we increase that COD to overcome and get activity, basically. So know that across the board, we found points where we could get at least a 2-log inactivation for all scenarios investigated. So I'd say that's a good thing for the industry. And many times we got far greater than 2-log inactivation at the end of this 60-second time period. So here's our sugar toxigenic E. coli data. And looking at salmonella, again, same exact setup they were seeing there with sugar toxigenic E. coli, we saw very similar trends. I would say that these are very reproducible. And again, we have in all instances, even at the higher COD levels, where we can achieve a greater than 99% inactivation of our target organism within 60 seconds. Listeria is the one where we had a little bit harder. And like I said, this Listeria data set was very reproducible. With the concentrations we were using, we were only achieving a 2-log inactivation across our Listeria data set, and it wasn't really driven by COD level at 80 parts per million PAA, whereas we saw very similar trends for chlorine and Listeria that we saw for our gram negatives. So to recap on chlorine and PAA, the concentration of free chlorine or PAA to achieve at least that 2-log inactivation, which I am not saying that's your safe harbor, but just to try and equivalent set across the board for comparison purposes, was 30 COD of 30. We had 30 and 80 parts per million of free chlorine and PAA respectively. And the 80 parts per million was really across the board once we looked at all three of those organisms. Know that we were also at 30 parts per million free chlorine at the mid-range COD, and at 2,500, we had to step up to 50 parts per million. So know that most implant COD observations were around 500 in this commodity. This can vary significantly based upon flume management and soil load of the crop, as well as if you have decay, if it's a storage crop. So know that just because they were this in Washington State apples does not mean they're going to be the same in your commodities or in your situation. And I would encourage you to find out those values. Listeria was a little bit more resistant to inactivation with PA under the conditions that we evaluated, but combinations of free chlorine or PA were determined, which reduced to at least that 99% mark for all 3 organisms that we evaluated. Getting in very briefly and looking at objective 4.1 in contact, and the students that conducted work in my lab on this was Rusha Borkar. She worked extremely hard to look at taking this work further and applying it to a vegetable commodity. We used slicing F1 hybrid cucumbers within this system and did an immersion type wash. We looked at reduction of salmonella or E. coli TVS 353 as a surrogate on inoculated cucumbers to look at cross-contamination and actual reduction on the inoculated cucumbers. So we looked at free chlorine or PAA in these concentrations of 0 as our control, 20, 40, or 80 parts per million, and COD levels of 300 or 2,500 milligrams per liter. We also tested those same physicochemical parameters that we had mentioned previously. And what we did was we took one inoculated cucumber and essentially intermingled it with 9 other uninoculated cucumbers. We then basically looked for the populations that were surviving on that inoculated cucumber and then looked for what had been transferred to the 9 uninoculated cucumbers, and as well as a 100 ml sample of water from that system. All of this was through a 1 minute exposure through this wash system. And know that we did negate the effects of the sanitizer by using sodium thiosulfate, which was effective across the board, as well as a little bit of a surfactant to hopefully remove the organism if it was present. Know that we did go to a 5-tube MPN for salmonella, and we used the Colert quantity trace so we could get the lowest possible quantification across the board. So let's get into some of these numbers. So similar to what Michelle was showing you, that on the actual inoculated cucumbers, we can also get log inactivation. So we did not see any significant effect of sanitizer here. So we've just merged them together. And you can see here that at a 20, 40, or 80 part per million concentration for chlorine or PAA, we were somewhere above a 2 log inactivation. And that was significantly different from our controls here that were set at 0. So we can see the benefit there. I know that we had a question in the queue, what is water doing here? Is it an antimicrobial? And the answer is no. What I can say is it's physical removal at that point. So really here we're seeing less than a 1 log reduction from that physical removal on that cucumber. So now I'm taking you to the upside down, right? Where we're looking at transfer to uninoculated cucumbers. And this number is a log % transfer. I've put in blue boxes to help with your understanding here, but know that across the board, that when we have our no sanitizer right here, we see very limited efficacy and we see a lot of transfer going on, right? So we're basically getting somewhere around 1 in 1,000 for every 1,000 cells of our pathogen we have, we're getting 1 transferred in this system because it is a dilution effect, right? That is going on if nothing else. But we see far greater efficacy if we have any amount of sanitizer present, somewhere around 1 in 10 million cells, right? And that speaks volumes to the benefits of using this and the real benefit of preventing cross-contamination. If we looked into E. coli transfer to uninoculated cucumbers similar to salmonella, we do see very similar trends across the board. The one standout is here at chlorine 20, where we did not get as great a control at the lower concentration of chlorine, specifically in our high COD level in this instance. Know that all of these experiments were replicated 6 times. So that's 54 uninoculated cucumbers for each organism tested in concentration of sanitizer. So we had quite a lot of cucumbers that we evaluated in my lab. And this was, again, very reproducible. And we actually look at the transfer to water again, we see our benefits here. So that if we don't have any sanitizer present, we're definitely getting that transfer going into water somewhere around 1% or so. And if we come down, or excuse me, 10%, sorry. And if we come down here, we're really, again, somewhere around 1 in 100,000 cells to 1 in 10,000 cells if we have any amount of sanitizer present. It was interesting to me that when we analyzed this data, there wasn't any effect of COD or sanitizer type. Usually we did see that effect of COD level, but it did not come forth. And I'm assuming just because we're giving it longer interaction time, right, that we're getting a little bit more benefit in separation there, or grouping there. Similarly, if we look at E. coli and that 20 part per million concentration, we did not see as great a control there. But again, once we get above 20 to 40 and 80 parts per million coming in at really good control levels. So for our key takeaways, we have low rates of salmonella transfer to cucumbers and high or moderate COD levels and properly managed. E. coli TBS 353 behaved similarly, and if it didn't, it was more conservative as a surrogate. So no, this can help further establish it as an appropriate surrogate for these systems. And I would just encourage you to start to assess your post-harvest systems. Know that ultimately studies like this can help support your decisions. Look for other systems. I know we haven't covered a lot of ground here, like hydrocoolers and a lot of other types of systems. If you don't find the data there, verbalize it as a high priority, right? So make sure it's on one of the researchers' lists that your commodity group commonly works with, so that we can start to collect this data for you. Ultimately, I would encourage you all, if you don't know for your recirculated systems how COD is looking, know that I would start to get an idea about what those values look like for you, in addition to other parameters that we've mentioned previously. And that'll give you a good basis from which you can start to assess how these systems actually resemble my own. I would be really remiss if I didn't include a little bit of other work from Contact. So know that we do have another paper that was published looking at spinach from Dr. Rohan Tidakar's lab, also from the Contact group, that we will put for you in with our post-webinar information. Last thing, I promise before Laura pulls me off here with the cane, we do have a post-harvest water a survey open right now. You can link to it right here. We'll also put the link in the chat. It will be open until May 15th. Many of you have already participated. Thank you so much. But if you haven't, please, we do want to hear from you and how you're managing your post-harvest water systems. So thank you very much.